IEEE, pardon, İngilizce geç. Hello everyone, welcome to IEEE Turkey section, the undergraduate academic conference. This is day 2. My name is Nezih Topaloğlu. I'm a faculty member in the Department of Mechanical Engineering at Yeditepe University. Today is the second day of the conference, and in the morning session, we will again have nice presentations from the students. But at the beginning, we will have again an invited speech, invited lecture. The lecturer is Professor Çağlar Elbüken. He's attending this conference from University of Oulu in Finland. Let me briefly introduce Dr. Elbüken. He got his bachelor's degree from Bilkent University Electrical and Electronics Engineering in 2004. Then he did his PhD in Mechanical and Mechatronics Engineering from University of Waterloo in Canada. Actually, he's I did my PhD in Canada as well, so he's my colleague from there. We had a nice time in Canada, good old days. And his PhD topic was magnetic levitation of microrobots. Then after 2 years of postdoctoral position at the University of Waterloo, he worked in Abbott chemistry as a senior scientist in the point of care diagnostics and R&D departments. Then he got back to Turkey and worked in Koç University as a research assistant professor. Then he started working in Bilkent University as an assistant professor. Now he's currently a professor in University of Oulu, Finland, in the area of biochemistry and molecular medicine. And his work, his research spans lab-on-a-chip systems, two-phase flow, viscoelasticity, nanoparticle synthesis, and purification. This is a very brief summary of nice works and nice career that he's making. And his lecture today is going to be about droplet-based microfluidic systems and their applications in biotechnology. Dr. Elbüken, I think I can now give the microphone to you, and you can maybe start. Thank you.
Thank you, Nezih Hocam, Murat Hocam, for the invitation and introduction. Just before we get started, do you see my screen now? I shared the PowerPoint slide.
Yes, Professor, perfectly.
Thank you very much. Okay, and again, thanks for being here. Sunday, 10 a.m., such a commitment to science. So it's also a pleasure for me to give a presentation in IEEE seminars, being a researcher originally from electrical engineering background. And the way I will tailor this presentation is also a little bit geared towards engineers, especially electrical engineers. So I hope that will be also useful for our attendees. And my title today is two-phase flow, or in other words, droplet-based microfluidic systems. So in general, this is a little bit maybe geared towards fluid mechanics and mechanical engineering, but as you will see, we have been working on these systems at system level, which requires also a lot of electrical engineering work that is integrated in these systems. And I will try to give you a flavor of the topic first. So if you haven't heard about these before, don't worry about it. We will just go over this in a few slides. And then I will also tell you about what the state of the art in the system and what are some potential uses of the technologies that I will be talking about. And so this slide is indeed a summary of my CV, in a sense that we have witnessed this revolution at small scale, first at micro scale, and then happened at nano scale. And it all started with this, the development of the first transistor. So it was a need in telecommunication. And there the engineers have, or scientists have, also understood the motion of electrons. So that was the deal. This is 1920s, 30s, 50s. And then the next jump in the technology came with this motion of these solid parts. So it's not the electron movement only, but also mechanical movement of parts, which we call systems as micro electromechanical systems. So again, this is beginning of 2000s, 1990s, etc. So there's again another jump in technology, and these devices are now in our pockets. Many of them exist in our pockets, in our cell phones as sensors today. And the next jump was to control motion of fluids. And this came in under the name of microfluidics or lab-on-a-chip systems. But really the technology was, we had all these microfabrication understanding and also the infrastructures all across the world. So why don't we start manipulating fluids as well, in addition to mechanical parts, in addition to electrons. So that's, in a way, it's all about the motion of material. And this basically summarized my CV as well. So my bachelor electrical engineering, then a little bit mechanical engineering, and then biotechnology. So now if we look at some of the subdomains of these microfluidic or motion of fluid systems, a subclass of those systems is these two-phase flow systems. And you are looking at right now one of those systems. You see some channels with widths of 100 micron width. And you see these tiny nanoliter volume droplets that are being generated. So that's what we mean generally by a two-phase flow system. We have two types of fluids. You can think of them as oil and water. One of them is dyed, so you see this as green or blue in this diagram. So let's say this is water. And this transparent section in this channel, up and down in this configuration now, is pumping oil to the system. So right now oil is coming from the left-hand side. And what is happening here is they don't mix to each other. So you basically form a fluid compartment into another fluid. So that's what we mean by droplet fluidic systems or two-phase flow systems. We have two separate phases that don't mix with each other. And if you look around, these systems are very heavily used. So this is the basic building block of these inkjet printers, or on a rainy day, that's basically what you are witnessing. In air, which is a fluid, you have water droplets. And again, if you look around a little bit, it has been used a lot in marking that you see on these aluminum cans, for instance. It's like coming in a dotted way. So that's a good indication. And when you see these, for instance, during COVID-19, this is something that we discussed a lot probably, that is everybody is generating these droplets. And when we look at the other fields of areas, biotechnology is using this very commonly. So we have these microvalve plates areas which compartmentalize the fluids that are being used. So it's not on the format as we discussed as droplets. So this is more conventional format of using these fluid separation and fluid compartmentalization idea. And if you think of in the field of, let's say, biology, chemistry, or biotechnology in general, the stereotypic image of those fields is somebody doing some sort of pipetting. So basically, it's all about manipulating fluids at small amounts. And as you can imagine, the benefit of small amount is the cost is reduced, so you don't need to spend a lot of samples. And interestingly, in many cases, you don't even have access to a lot of samples. So if you're working with some precious bodily fluids, or let's say nucleic acid samples, then the amount of fluid that you have is also very limited. So that's why these tiny droplets have a strong technological benefit. And if you look at the properties of these systems, these have some unique properties. These type of droplet fluidic systems are quite unique in the sense that we can generate these systems and we can split them very easily. So when it requires, we can combine different types of droplets. If it is needed, we can incubate them as large numbers. We can make sure they go through different processes, or we can look at the interface of them. So it's also possible to study interface chemistry with these systems. So in a way, I will just put a bold statement saying that most of the operations that you do with a pipette, or indeed all of them, is existing as technology in these droplet fluidic systems. So we can summarize it in a way that anything that you do with a pipette in a biology chemistry lab can be transferred into this format with much smaller volumes in an automated manner. And that gives you an upper hand in any of these demanding assays. So if we look at what we have done in this field, so this is a summary of our completed works. So we have studied on understanding the formation of these systems. So how these droplets are formed, what are the dynamics here. Then we have, after we understood this, we try to understand once these droplets are formed, if they are sent in a network of channels, how do they behave? How do they go? So is there any patterns that we can observe? And I will come back to this topic at the end of my seminar. And while we are doing that, we realize that the field lacks some sensors, etc. So we developed some electrical sensors to sense them, to first count them, and then to detect their material properties. And once we have all these building blocks, we also made some whole blood assays using these droplet fluidic systems as well. So this black liquid, for instance, that you see here is a whole blood sample that is introduced into these tiny liquids, which are on the order of, in this case, I think it's around 100 picoliter volume manipulation that we can have. And I will tell you a little bit about some of these technologies, starting from the sensors, since I think this is also a little bit more electrical, and you can see the simplicity of the system and the difference that it made in the literature. So what you're looking at here, for instance, is one of these fabricated devices. So if you are wondering how they look, this is fitting into your palm. There are different ways to make it, but the most common one is using some sort of polymer glass systems. And inside these systems, there is these microfluidic channels that are transparent, so not visible in this picture, but that's the pattern that we are looking at. And these black, or the metal parts that you see here, the shiny parts, are some electrodes. And on the upper picture, you see that this is the channel that comes and then splits into two, and then some black or non-transparent electrodes are occupying the screen on the top. And what you're looking at is indeed some sort of a very simple capacitive sensing that is done in these systems. And because, again, these are usually systems made out of oil and water, there's a huge contrast between dielectric constant, and that gives us a very good change in capacitance if you measure it using some coplanar electrodes deposited under or over these systems. So then using those systems, we can detect droplets, and using two of those capacitive sensors, if you just make a differential measurement, as it is in this case, you can say which way these droplets are going. So that's something that we indeed first did in this field when I started working in this field 15 years ago. So this was my first study. I normally don't present this too much because it's a little bit old, but interestingly, we went back to this topic after 15 years, and I will tell you a little bit about that story as well. So when we start talking about applications. So then basically with these systems, again, by using some simple sensors, off-the-shelf sensors, a little bit of coding, we can have these systems where if you have a large fluidic network, so let's say you are designing a relatively complicated assay using droplets, you can monitor where all your droplets are going. So what you're looking at is basically these up peak represents a droplet going up, down peak represents a droplet going down. So at every junction, what is happening with these droplets and how they are manipulated. So that's the user interface of such a system. Now, another example that we have completed is these whole blood assays. So this is, here we have just implemented a hemagglutination assay. It's a very common assay, again, in clinical settings. So what we showed here is a prototype, a proof of concept hemagglutination assay. So basically red blood cell agglutination and sedimentation assay inside droplets using whole blood. And you see the different features of the chip. So let me just run all the videos. So again, this is the chip geometry that we are looking at. So we can generate them. Once we have a droplet, we can mix it with other fluids. And this is indeed ensuring a complete mixture. And we can also do electrical detection with these systems. So that shows you some of the building blocks, but also built around a complete assay, which is, again, already being used in clinical settings. And if you look at the whole system, what I'm showing you is, of course, some microscopic, optical microscope images that is looking into certain areas of the chip. But of course, there's all these peripherals, especially when you start doing some sort of detection and manipulation using electrical systems. So there's also some systems built around this. So if you want to turn this into an equipment, this can very easily be a benchtop equipment, which can be used again for different types of assays. So one of the most fundamental problems in this field was how to make these droplets consistent to each other. In other words, can we keep the volume of these droplets the same while this formation is being happening? And that's, again, a problem that we tackled a few years ago, and I think we have a good solution now. So that's something that we call as droplet monodispersity, how to make sure these are the same size of droplets. So in that case, what we are looking at is some very simple equations, empirical equations exist in the literature that gives you the length of these droplets as a function of the flow rates. And what we did was indeed very simple. So some analogy to electrical circuits. Again, it's interesting. So if you look at the electrical circuit curriculum, in first year, when explaining voltage current relationships, the analogy is given from the fluids. So here I'm just doing the opposite indeed. So assuming we know good electrical engineering, so we can use those electrical engineering tools to understand what is happening at the fluidic level. And because this is two-phase flow, it's a little bit of a more interesting problem because we have these interfaces and Laplace pressures that normally don't exist. But we can still use these electrical circuit analogy in a way that if you run the system with a syringe pump, which is basically just pushing a syringe, so it sets the volume or flow rate to the system. In other words, the equivalent of that is a current source. Whereas you can just pressurize the system, you just put some fluids up and then you make a tubing connection because of this hydrostatic difference. That will just also pump the fluids, but that's a basically pressure-driven fluid. So then the equivalent of that is a voltage-driven, voltage difference-driven electrical flow. So then you can use these analogies, and then when we started to just write down the equations, very simple, equations that you have all seen in the first year, we have realized that in order to get this length of the droplet, which is correlated to the volume of the droplet, equal, and I'm very sorry for the background noise. Let me see if I can do that. Anything to improve it? Okay, that maybe helped. So when we do this analysis, we have just written down this correlation between the flow rate ratios. And we have seen that in a pressure-driven system, because of this junction pressure, which is the junction voltage in this case, it keeps fluctuating because as these droplets are being formed, this Vj is changing. And a simple solution that we offered was, okay, why don't we just supply them from a single source? So then this term completely cancels even if there's a variation is junction voltage or junction pressure, because then everything is related to the resistance ratio, which is fixed in the system, even if it is a two-phase flow. So our simple solution was, if you would like to run these systems, they have to be supplied from the same source. So very simple, but then we extended this system to also understand this Laplace pressure and also this two-phase flow. Again, this is something that doesn't exist in electrical circuits because it's just basically electron flow. So it's one-phase flow, if you can just think of it that way. But here we have two phases. So imagine that electrons and holes are also carrying the flow simultaneously in the same direction. So that is the analogy that we can use here. So then when we model these circuits, we use some SPICE software also to understand the dynamics. And there are also a few design rules that we have given. So this was just published last year, and we have shown that we have the world record monodispersity. So coefficient of variation is a measure of monodispersity. The lower, the better. So as you can see, it was around maybe a few percent, 1 to 3%, and we were able to go down to an order of magnitude. And as you can imagine, at this level, an order of magnitude improvement is quite significant. that we were able to get the most monodispersed droplets by using some simple electrical circuit analogies. So now you are probably wondering, okay, what are some real uses of the system, of these systems, and are any of these technologies being used? Now, let me show you 3 applications only. And probably you'll be more familiar to the first one, digital PCR systems. So we have heard about PCR in the last few years, thanks to COVID, that it's a technology used to detect basically nucleic acids, amplify nucleic acids. And DNA, RNA-based detection systems use this very, very commonly. So digital PCR is a new technique that uses these droplet systems for absolute quantification. And I will tell you a little bit more about this. And the next application in the middle that I'm showing is for single cell profiling. So let's say you get a tissue sample and then you are trying to understand the RNA expressions in that tissue, or even just, let's say, cell types, how many different types of cell that I have in a given tissue. So for these systems, you can just compartmentalize individual cells into individual droplets. And then again, each of these droplets is working more like a single pipetting experiment that allows you to do this assay. And this can be, again, automated in this droplet format, exactly like what I showed you in the previous videos. Another common example is protein crystallization. Again, a very, very commonly used technique in, I would say, biochemistry. So to find optimal conditions for protein crystallization, again, these type of small volume droplet systems are used, and these are all commercial examples. So these are not articles only. And if you look at the first one now in a little bit more detail, digital PCR. So we all know what a PCR is, I guess. So it's basically just amplified a piece of nucleic acid, DNA or RNA if it is reverse transcripts. So then the problem here is after you amplify it, in order to detect the amount of your target sequence, you have to just do some fluorescence imaging. So you need a calibration curve, and the fluorescence intensity you get, you have to just compare with it. So it's not very precise and then requires calibration curve. Whereas in the droplet PCR field, you take the sample, let's say a blood sample, you divide it into these very, very small volumes. It is so small that in each of these volumes, you either have a target molecule or not. So think of it this way. So you just take your sample. This is your original sample, let's say your blood sample. You divide it into small volumes. So you end up with these volumes which have a target DNA or zero target DNA. So you just amplify it, and the number and the intensity does not matter at this point too much. So it should be amplified enough so that you start seeing some of these droplets to give fluorescence signal. And you don't care about fluorescence intensity anymore. So this is intensity independent, because as soon as you are above a threshold, you say, okay, there was one target at the beginning. So I can count this droplet as 1 and the next one as 0. So that's why it's ones and zeros, hence the name digital PCR. So by basically simply counting the number of droplets in these systems, which are bright, you can say, okay, at the beginning I had 1, 2, 3, 4, 5, 6, 7 target molecules in my sample. So it's a very, very precise technique. But as you can imagine, it is susceptible to changes in the volume. So if your volume is not consistent when you do this partitioning, it is quite likely that instead of 1, let's say for a larger droplet, you may have 2 analytes at the beginning, and eventually you will count this as 1 again. So that's basically the imprecision in the system inherent because of the variation in the volume. And that's also shown, has been proven with many biological assays that this is the one of the most tricky points here. So basically that monodispersity that I mentioned to you is extremely critical in these systems where we need to make sure that our partitioning is the same volume so that we get some very precise measurements. So as you see, even with some simple engineering techniques, we can help the field improve in its precision. So now I will show you some other techniques, and this is a little bit maybe about material science. The application may not be that relevant, but again, I will show you the technologies that are mostly, again, coming from some engineering background. So the next thing is, for instance, real-time impedance analysis of droplets. So this is some system that we have developed a few years ago and published 2 years ago. Here we were able to do that type of analysis, again, electrical sensing using some coplanar electrodes under microchannels. But this time we extend the capabilities in a way that in real time we can measure size, volume, dielectric properties of these systems. And this is a completely automated instrument that we developed for a material synthesis problem. Because when you start running these systems, let's say you form these droplets and you will synthesize some materials inside them. Think of these again, some biochemistry tubes that you're manipulating. So we need to make sure that the system has reached the optimum condition before we start any synthesis. So that's why this real-time detection ability was pretty important. And this time we used 3 coplanar electrodes under the channels. It's shown by these gold rectangles, and these blue droplets are going in the channel. So you're just looking at a side view of the system. So these are squeezed droplets. And by doing some real-time signal processing, we were able to measure some very precise parameters of this droplet. And you can even extend this to get the shape of the droplet, these caps, which will also tell you a little bit about surface tension or interfacial tension. I thought that was a little bit advanced at that time, but we left it at the point that we can measure dielectric constants, conductivities, and also the size of the droplets. So now this is a software freely available. So if anybody is needing it in the field, and what this enables is, let's say for some sort of a material synthesis application that you want these content of the droplet in specific format and chemistry, and then you can turn on your synthesis. So with this system, you can do this real-time detection and also synthesis after the optimal conditions are acquired. And also for material synthesis, we use this for many different types of materials. The monodispersity, or the quality of the material that you're producing at microscale, is extremely important. So with our techniques that we can ensure monodispersity, and also we can do synthesis when the system is ready. And what you're looking at is some example synthesis results. So these are just some silica particles that are, again, very commonly used in, let's say, chromatographic columns, etc. And if we use the conventional systems, as you see, the sizes vary. With our technique that I mentioned you at the previous slides, we can bring down the CV significantly, and then we can ensure in real time that this is the monodispersity is achieved in the system, and then we start the material synthesis, these steps. And I don't explain the chemistry here that much. I don't think it's not too relevant for this meeting. But as you can imagine, any type of chemical synthesis that you do can be automated in these systems, and then you can do them at very high precision in an automated system at high throughputs. And if you are wondering how these systems look like, this is just one video that will show you how it is being done. So you have some fluidic connections here into the system, and then in real time, you're looking at the microscope view. As you see, microscope is sliding, so you're just looking at the droplets. Those black lines are the electrodes, so they're connected to an external lock-in amplifier system with a custom software that is analyzing these droplets. And then once you're happy with what you produce in the system, you can also transfer them into vials, to the external world as well. So they are not destined to stay in these microfluidic chips. So now you see some sort of microparticles. It may not be very clear on your side on the video, but those microparticles or micro droplets being transferred into the tubing. So that's how it looks. And we have done it with different chemistries. So this is quite new, unpublished data with Professor Tuncel at Elbüken University. We have also applied this with some capsule formation. So it's not just the surface, but also the content we can also vary. And then these are very commonly used again for drug release type of applications. And we are currently investigating the properties of these capsules and also the monodispersity. So you're just looking at some optical microscope image of these systems, which shows these systems, and you can see how precise the formation is, and any defects, let's say in this region, maybe some droplet being a little bit smaller, is very easily detectable when you put them as a monolayer even on a glass slide. So you can see the precision that we can obtain with these systems. So these are indeed some of the completed works, and now I will tell you about two more projects in the maybe last 10 minutes or so about what we are currently working on. So another very important idea in this field is, well, now that we have this precision in manipulating these fluids at that level, so can we use it to understand biology a little bit better? And the question is originating from the similarity between these systems and cellular systems. So if you look at a cell, for instance, it's a bilipid layer, let's say some blood cell that we are talking about, some human cell. It's a bilipid layer stabilized, and then inside it there's a factory going on, all sorts of biochemical processes. So, but inside it's primarily aqueous, and outside it's an aqueous world. So we can make such systems in a very controllable manner, and we can control what is on the surface, what is inside it, and what is outside it. So then the question was, well, can we start doing biology from scratch? And the keywords here in this field are artificial cells, artificial organelles, lipid protocells, and another approach that is also quite popular today is synthetic biology approaches. But this is a little bit of top-down approach, whereas in artificial cells, artificial organelles, this is a bottom-up approach. So can we try to understand biology by building it in the laboratory from scratch? So this may again sound like these Miller-Urey experiments, 1950s, I think, but it's a little bit different. It's not just giving the energy to the system and then expecting some organic molecules to emerge or some reactions to happen. Instead, we exactly know the ingredients of a cell. So can we put it intentionally into a system so that we may be able to make a cell? So it's right now the field is, of course, as far from the expected results, but it's developing very recently. So again, the idea is compartmentalizing these chemicals and also the contents of these cells. And if we have this precision that we need, can we build some very simple cellular functional building blocks on these systems? And this is something that we have been, we started working on for the last maybe 2-3 years. And then with all this understanding that we have previously with our manipulation abilities, our goal now is to make these cell-like structures and also to let them self-assemble. So that's one of the things that we're looking at. And again, from an engineering perspective, you can just think of this approach as when you learn a new software language, that's probably what you do. First, just typing hello world, and then you start building on it. So that's basically where the field is right now. So we know how to make these compartments. There are some simple building blocks that are already implemented. Of course, none of them is as complex as a cell, but that's where the field is. So we were able to type hello world to the screen. So now the field is evolving, I would say, a very rapid phase. And one of the dreams in the system is once we make these some simple cell functions, can we also start implementing these organelles? So in other words, some compartments into another compartment. So a cell divided into different compartments. And those type of things, again, if you just look at the fluid mechanics of it, just compartmentalization idea is not super challenging, and we are able to do those systems now. So we can make these single bilipid layers or multi-layer systems. Again, for drug release, this is very relevant, as you can imagine, because it allows you to control how fast your cargo is released to the environment. Or we can make these compartment inside another compartment. So in a way, these nested compartment systems. So these are all quite possible. So that's basically what we are looking at, mostly about formation, but there's a whole lot of research going on, especially this time from the side of more biology and chemistry people trying to implement their know-how and then replicate properties and functions of cells in such systems. So this is an ongoing project for us. Another thing, this was also very much related to the beginning of my talk, was I was telling you about these types of networks and how this bifurcation is happening. So one problem that we are looking at now is in a bifurcation network, if it is a natural system, there's probably some sort of material transport happening. So what I'm showing you here is some sort of examples of that. So in blood, for instance, it's basically oxygen transport through red blood cells in plasma. And we have many examples of these bifurcating networks in the nature. And we can also build such systems in our buildings. For instance, we have to transport fluid, but in this case, this is mostly about just the transport of the fluid. So we don't transfer particles, for instance, in this case. Although there are some examples, it's quite rare. In nanofluids, for instance, that is the challenge. But in most cases, in our engineered systems, we just deal with bifurcation of the fluid, which is a well-known problem and then a well-studied problem. So we know how to engineer these systems. But once we have these discrete particles in the system, which may affect the system dynamics, the problem suddenly becomes very interesting. And as you can imagine, this is one of the fundamental things in our body, this oxygen transport through red blood cells. And at every bifurcation, these red blood cells have to either go right or left. And then that determines how much oxygen this downstream tissue is getting. And all these diseases, cardiovascular diseases, for instance, is very much related to these ones. So any resistance change here, again, atherosclerosis, would lead to, let's say, less oxygen transports than that tissue not being fed properly and then dying out. And then this is basically an engineering definition of myocardial infarction. So the question that we are asking is, can we understand this type of particle transport in these systems where particle has a significant effect on the dynamics of the system? So if a red blood cell going here, it is changing the resistance here. Again, think of it like an electrical circuit. Then that would affect the next red blood cell coming to the stream. So that was indeed the problem that I mentioned to you as my first project 15 years ago, these droplets going right or left in these networks. So can we understand? So we basically use these droplets as an analogy to mimic these systems. And I will show you one video, for instance, some droplets coming from up and then they go left or right. And then our question was, if we watch this long enough and if we design this system, do we see a pattern here? So can we really precisely control, let's say, the oxygen transport to the downstream right, left tissues? And when we do this, we have some idea about what is happening. But once we started to dig into deeper in this, we realized that this is a very, very complicated problem. So now I will show you some numerical results. Again, that system that I showed you, droplets coming, going right or left, is shown in these nested geometry. And what we are looking at is really downstream. So this is away from the heart at the tissue level. Do we see a pattern? Do they go equally? And of course, there's no noise here, no uncertainty, etc. This is a very, very ideal scenario that we are trying to simulate. And what we realize is something very, very interesting. So when we run the system, everything under exactly the same condition, except we change the incoming droplet frequency. So how fast your heart is pumping. So how fast we send these droplets. So if we send them at a certain frequency, we see that these droplets go right or left at the junction. So it's very ordered. So that's the pattern. So we can see a pattern. When we increase the frequency to, let's say, 2.5 Hertz, we see that there is a pattern. It's a little bit longer, but not so difficult to detect. So right, left, right, left is the pattern that you see at these tissue level, second level junctions. But in between 2.2 Hertz, we haven't seen any pattern. So what I would expect normally is maybe a pattern, but maybe something like length of 4, I would say, would be very reasonable, but we haven't seen a pattern. And as I said, there is no experimental error or anything here. These are completely deterministic system that we just coded in on a computer. So there's no uncertainty that we are talking about, maybe except rounding errors, but we have verified that it's also not the rounding errors here. So then the question is, what is happening here? So there's some order, but in between some disorder. And again, this pattern just repeats itself, so it's not specific to around 2 Hz. So what we came to conclusion is what is probably happening is, again, a chaotic system that we are looking at here. So I'm not sure if you are familiar to this plot, but this is a very famous plot called the logistic map that is used very commonly to explain these chaotic systems, and it's a bifurcation diagram at the end. So it tells you the system dynamics. So it's basically a snapshot of how a system behaves at all the frequencies that I showed. And my frequency in the previous slide is equivalent to this R factor here. So this has been originally developed, this plot for population dynamics, how population change. And in our case, we were able to just see a very interesting analogy between our ordered sorting versus chaotic behavior. And that's exactly what you see as you change R. You see the system going from very deterministic to some random-like pattern. And that's basically the signature of chaotic systems. So we have seen that we have obtained such systems, very similar systems. And now we are basically looking at how these bifurcation networks are happening in the body. So this is some, we are looking at vascular networks, urinary networks, and we are trying to understand the relationship between these particle transport and how these networks are being formed. And we are using some very simple experimental models, some chicken egg with some blood cells on it. Again, as the embryo is growing, you can see that this is almost like a high school experiment. This heart is pumping and then red blood cells moving. So we are trying to understand whether that motion can be understood. And the next question is basically, in the body, can we have such level of precision to understand what is happening in the cardiovascular system? And now we are also combining this with some microfluidic systems. So we are trying to understand how these blood vessels are being formed and how this transport is happening. And this is a super complex system we are dealing with, but the analogy that we are using is these two-phase flow and also discretization type of ideas. And with that, I will end my presentation, and I would like to also thank all our researchers that we have worked together, our collaborators, funding agencies, and to you for your patience on a Sunday morning listening to me. And if you are interested in these systems, we also have some positions available, so PhD positions. I think that time was beginning of September for application. So please let me know if you are interested in working these systems. And with that, I will pass it to Nezi Hocam again.
Okay, thank you, Çağlar Hocam. That was a very interesting and very inspiring talk. Droplet-based microfluidic systems, and at the end, it was also interesting that we have, you started talking about chaos. I was also surprised that because it's like you had these bifurcation, let's say channels being bifurcated, and at the end, we reached on this bifurcated diagrams. So that was from physical to mathematical passage that I have seen some sort of. I don't know if I was able to explain it well, but so there are some couple of questions regarding this chaotic thing. I would like to ask something. So do you then claim that the our blood system is chaotic because probably that's intermediate frequencies are going on there? So or do you think it's kind of, or from our sense, is it chaotic?
Yes, so our hypothesis for that project is still, I would say, on the air, but we cannot claim anything about blood at this point because it's probably much more complicated than we think. But what we are claiming here is if you have a bifurcating network and if there's a transport happening in these systems, and if you avoid any collisions between these particles, as I showed you in these droplets, then that very simple system that we also modeled experimentally is behaving chaotically. So then if you make the system even a little bit more complicated, then it makes sense for us in the sense that, oh, it's probably more chaotic. But what the type of chaos we are talking about is a little bit, our terminology, our daily terminology might be a little bit misleading. So that's why I don't attempt saying that, okay, blood flow is a chaotic system until we prove using the mathematical tools of chaos theory that red blood cell movement is chaotic. And the difference right now between our system and the real biological system is density of the materials. So this density of these droplets, and also the collisions, which is again related to density. So we assume there is no particle-particle interaction in the system. And that's how we simulate, and that's how we experimentally mimic the system using droplets. So these neighbor droplets don't talk to each other. But in case of droplets, we know that that's not, sorry, in case of blood, we know that's not true because these red blood cells, they just fold onto each other. They are at very high concentration. So it's probably a much more complicated scenario. So we basically start with a very, very simple version of the real scenario, which we claim to be chaotic at this scale.
But the simulations that you were making was fully deterministic, right? That's what you said. It's not like a Monte Carlo simulation or anything.
Yes, and indeed, they're fully deterministic. And if you look at the type of chaos that I'm claiming here, that logistic map, that's also fully deterministic. So maybe if I just back up a few slides. Yes, so that's this type of map is coming from this equation. So very simple equation, deterministic. You just type it in a computer, and then you get also chaos. So chaos. The chaos that we are talking about here is basically also deterministic. So there's no stochasticity here.
Okay. Another question is, these droplets in droplet microfluidic systems, we know that in when we're talking about conventional microfluidic systems, especially where the flow is usually laminar and mixing is a problem, right? I mean, you need to use some sort of mixers or some other approaches. Maybe active mixing technologies, etc. So what is that case in droplets? I mean, for example, you have a droplet that holds a species as something, I mean, a molecule, whatever, and another type of droplet coming from another source, and you need to mix them. Is it easier compared to conventional microfluidics?
It is much easier, and it's a very good question. I wish I could learn the source of this question. Is it from the students?
Actually, it was from me.
Okay, from you. All right. Yes. Yes. Very good point, Nezi Hocam. So it's almost to the point that this is a schematic explaining your question. So if you have a normal single phase system, and if you run the system, we know that there is dispersion happening because of parabolic flow profiles. So it's difficult to keep the integrity. And if you even want to mix two fluids, that's a difficult problem because of laminar flow. So it's maybe a subset of your question is here. So mixing only happens by diffusion, in other words. Yes. But if you compartmentalize these different types of fluids, let's say 3 colors of fluid in a single droplet, in order to mix, all you have to do is let it to turn some, make a right-left pattern. And what is happening here is basically this stretch and folding happening in vortices inside these droplets. And it's just like your car turning having a right turn, the inner wheels and then outer wheels go different distances. So these vortices fold differently onto each other. So if you do this right-left only a few times, that is the equivalent of this stretch fold action in a Play-Doh. So then even by diffusion, so the mechanism is not changing, the physics is the same, even by diffusion, because you have now a very, very long interface between the fluids, mixing is almost instantaneous.
Wow.
So that,
Yes.
that's very well
But
built in. And that's why indeed we have these serpentine channels in our systems that I showed with blood. So that's where we mix the fluids.
Yeah, okay, very interesting. All right, so we also have some questions from YouTube. Let me just read them to you. One question is about the PCR. If after amplification, more molecules can exist within the same volume, what is the probability of an uneven distribution of molecules initially? For example, what we mean by uneven is 10 molecules do not distribute in 10 partitions, instead accumulate in one. So I don't know if the question is clear to you.
Yes, I mean, this is a statistics question basically. So I don't have an answer up front, but these are all well modeled in statistical, using statistical methods. So the question, from what I understand, is asking, let's say, what is the probability of having not let's say not 10, but 2 targets in an individual droplet, and that's a function of the concentration of your target molecule and also volume. So you have to just basically have a rough idea about how much you have, or if you don't have, then you can do this in dilutions, and that's what's done usually. So you dilute your sample so much that you minimize the risk of having 2 analytes per sample. So that's a very well-known problem, and it exists even if you have monodispersed droplets, and that's basically the inherent statistical mistake that will exist in all these, in digital PCR method. So you cannot get around it even if you have perfectly equal volume isolation. So it's basically a statistics problem.
Yeah.
Which is modeled by Poisson distributions.
Oh, okay, Poisson distributions.
Yeah.
Taking place. Okay. All right. We lastly, there's a question from an electrical and electronics engineering student. Actually, this is an important question. Many people is probably asking. I will also note your answer with my pencil. He's asking, what are your advices to learn biological processes? Because for an EE student, it seems very confusing at the beginning when you read. So where should I start? He's asking.
Okay, this is just like these YouTube type questions. I also follow some great YouTube channels, and I always have this question in my mind. And these people talk about philosophy, and I have no idea about it. And
yes, where should I start?
How can I start?
Yeah,
it's exactly the question. So, well, I don't have a magic answer, unfortunately. But what I would recommend is, I'm sure there are some textbooks, you know, at starting level, etc. Again, because I'm not an expert in any of these fields, I cannot name you anything. But the way that I found the easiest is to just jump into it. And what you can do is, you know, if you start collaborating with these people, that's what I'm doing basically. So I myself, again, don't understand biology that much, but I developed these tools, and what I'm doing is just showing this to my friends in other departments. And then they really come up with most of these ideas, and then they get motivated about also integrating these technologies into their stuff. And along the way, I just basically learn what we are doing together. So that's my very simple answer. So you can just jump into it by putting yourself in a project that has some sort of, I don't know, biology, chemistry, whatever you are interested in. And that's basically some baby steps that you take in the fields. And I have a lot of friends also that completely changed their field of research after such baby steps in some different directions from their original discipline. And when you start doing that after maybe, I don't know, 10, 15 years, you will see that all these boundaries that we put between disciplines is also quite fake. I mean, we just compartmentalize certain topics and say, okay, this is electrical engineering, and now this is mechanical engineering, and oh, there's some separate building and then faculty on the other side of the campus, which we call biology. Okay, they're so different.
Yes.
Well, you see that it's quite fake indeed. These boundaries are just man-made boundaries. So, I mean, some fundamental principles exist It's just your hands-on skills. If you develop those skills, then you can start speaking different languages, really.
Yes, okay. And so your explanation also underlines the importance of interdisciplinary studies, and yes, project-based learning. Maybe that's what maybe you're also suggesting, because if there's a project that you are in, there's these specific topics that you have to learn and you have to improve. So yes, that's a very good point. Thanks for that advice as well. So I think this is the end of all the question session. I'd like to.
May I ask something to Professor Çağlar?
Yes, please.
I'm sorry. I think I'm taking advantage of being a host, in the meeting. That's why I didn't type on YouTube, because my account is going directly with IEEE Turkey. That's why I just switched here, Professor, with your permission. I'm very interested in this behavior, the fluid behavior, especially the one that you talk at the beginning and at the end, this, bifurcation problem and chaos. Now, I'm very interested in this motion, and because, we use I'm a signal processing guy and wireless communications guy. We're using this kind of models in the mobility models of huge mass plus individuals, especially the vehicular motion, because, there's a guided medium in here, and then basically cars, we can say that the vehicles are moving in the direction, some sort of, flow, probably maybe far from the fact that you guys are doing something totally different. But at the end of the day, we are trying to use these stochastic models in our systems to basically get some sort of ease on the theory. Now, are you guys interested in modeling the behavior of the motion? This is my first question. Or how much are you guys interested in? The second one is, because you talk about extremely similar topics that, as you said, in EE, especially communications, is there any sort of coding relation? For example, when you encapsulate something inside, okay, is there an easier way to detect it by a specific coding? I mean, like maybe size, or maybe the individual behavior, so that the detection is easier, or something like that. Basically, two keywords, the mobility behavior and coding, probably, since you are having this EE background, probably you know what I mean. I see, huge connection. I cannot verbalize maybe 100%, but I think, you get the idea, Professor. What do you think about those two?
Yes. Indeed, I'm also thinking along the same lines. So because we have probably the same frequency coming from the background. So the type of question that you summarized is, let's say, car networks in traffic network, let's say, is much more similar to our droplet systems than our droplet systems to the biological systems. Because again, in such a network, there is no collision. So you keep everything discrete, and then you try to minimize the particle interaction. So that's basically a very nice flowing traffic, a dream traffic, when there is no interaction. They don't even see each other. So this is how we approach the problem right now. And the droplet systems that I showed you is indeed a much better model to your problem. So our problems are very similar, whereas the biological problem is completely at a different level because of the difficulty there that I just explained. Also, Nezi Hoca's first question. Now, the type of coding that we use is, I would say, probably very similar. So the ones that I showed you here was from some MATLAB code and iterative modeling. And there are also some other software that is also available. And I think there are also very much similarities in telecommunication industry. So you send these pockets of information, again, compartments of information on a there's a spatial temporal relationship. So there's time and also space related. So it's exactly the same type of problem. I would assume that, again, telecommunications people are maybe much far ahead than us because we can have this right now all across the world online. So they solve most of their problems. Whereas we are at the beginning as our research group in this topic. But what we are trying to do is basically bring these three things together. So One thing is a real biological system, which is, we know, already very difficult. Can we mimic it in the lab experimentally? So that's what we call the in vitro replication. And there's this in silico version, which is the computational modeling. So these last two come together very nicely. We have already done that. But now trying to link this to a more complicated problem is what we are being challenged at this point. So that's why, yes, those type of coding is very heavily used, and my output was indeed something very similar. And I think there's a lot that we can learn from specifically telecommunication people.
I appreciate, Professor.
Yeah.
Okay. Thank you again, Professor Elbüken, for your time at the Sunday morning. It was a very useful talk for all of us. Thanks again, and I think we can keep on with our agenda. I think the next talk is going to be from Mr. Muhammed Yusuf Efe from Boğaziçi University, and the topic is a vertical federated learning method for multi-institutional.
We first close this session, then start again in 2 minutes.
Okay, so then we are giving a 2-minute break, it looks like. So I'm now closing the session. Thank you again, Dr. Elbüken, and we will meet in 2 minutes for the next section. Thank you very much.